Good afternoon, friends. At the outset, I must thank the organizer for giving me this opportunity to present my video recording of the paper which I would have shared and which I would have presented in Mauritius last year. Thank you for giving me this opportunity. I would like to speak on a topic which is very relevant as of now. The topic on which I am going to talk today is association of adiponectin gene with metabolic risk factors in postmenopausal obese women. I am Professor Sunita Tiwari, heading the Department of Physiology at King George's Medical University, Lucknow, UP, India. This slide shows King George's Medical University. I just wanted to make you people familiar with this majestic university located in Lucknow, UP, India. It is about 115 years old, established in 1905 by King George V. It has the building, as you may see, is beautiful. So is its reputation. It's one of the oldest and a very well-renowned medical university globally. Adiponectin. I would be talking mostly on adiponectin. My work is related to adiponectin. I would like to just apprise you a little bit more about adiponectin and why I have chosen this work. It is involved in a number of metabolic processes such as glucose utilization, fatty acid oxidation in muscle, decreased insulin resistance in the liver, and the metabolism of adipose tissue. Low adiponectin concentrations in postmenopausal women has been seen to be associated with adverse changes in the carotid intima media thickness and stiffness that is not dependent on other cardiovascular risk factors. Decreased expression and plasma levels of adiponectin may serve as a marker of increased metabolic and inflammatory risk. The potential role of adiponectin in obesity and related pathologies is directed mainly to protection against atherogenesis and insulin resistance. The visceral fat accumulation results in reduced levels of adiponectin, and it was reported earlier that centrally located fat was the main determinant of variability in adiponectin concentration in healthy postmenopausal women. As we all know, the prevalence of obesity has increased dramatically in the recent past few years. It is one of the commonest disorders in the postmenopausal women. Approximately 65% of the women are obese. Menopause contributes to development of central obesity, insulin resistance, and derangement of glucose and lipid metabolism. We all know that obesity is a state of chronic low-grade inflammation. It explains the development of obesity-related pathology. It plays an important role in the development of insulin resistance and the comorbidities, that is atherosclerosis, dyslipidemia, hypertension, prothrombotic states, and hyperglycemia. Adipose tissue is an active metabolic tissue. It plays a central role in the regulation of energy homeostasis. It secretes adipocyte-derived molecules including lipid metabolites and adipocytokines like adiponectin, leptin, resistin, TNF-alpha, interleukin-6, etc. The visceral adipose tissue accumulation is an important predictive factor of lipid, glucose, or atherogenic disturbances, while location of adipose tissue in the lower part of the body is not associated with increased alteration at the metabolic level. So now we talk about the different types of adipose tissue which we encounter. One is the subcutaneous adipose tissue and the other is the visceral adipose tissue. They show some functional differences. They have different capacities to produce adipocytokines and they differ in mRNA expression. VAT has a greater lipolytic activity than SAT, from which fatty acids are directly delivered to the liver via the portal vein. Increasing the delivery of lipids to the liver promotes triglyceride synthesis, and that exacerbates dyslipidemia. It has been demonstrated that the removal of visceral fat mass, improves insulin sensitivity. This does not imply that SAT, that is the subcutaneous adipose tissue, may not contribute to metabolic abnormalities. For a given adiposity, there is a large heterogeneity in the metabolic and cardiovascular risks, mainly linked to the location of excessive adipose tissue. Only a few studies investigated this relationship between the adiponectin gene expression with metabolic risk and insulin resistance. This study was designed to investigate VAT and SAT adiponectin mRNA expression in different adipose tissues, that is VAT and SAT, and evaluate its correlation with metabolic risk factors in postmenopausal obese women. Methods. A total of 68 postmenopausal women were recruited. 34 were obese, which were included as cases, and 34 were non-obese, which were controls. The fasting blood samples were collected and the abdominal VAT, visceral adipose tissue, and also subcutaneous adipose tissue were obtained after informed consent. The tissue samples were stored in RNA ladder for RNA extraction. Anthropometry, the parameters were body mass index, waist circumference, and biochemical parameters like plasma glucose, lipid profile, serum adiponectin levels were estimated. The plasma glucose, lipid profile concentrations were determined using an automated analyzer, and the kit used was of company Merck. The serum adiponectin levels were measured by ELISA. Total RNA was isolated using tri-reagent. RNA was quantified by measuring absorbency at 260 and 280 nm. Adiponectin mRNA levels were estimated by one-step RT-PCR, and the results were expressed in terms of normalized ratio of samples with calibrator. The data was summarized as mean and standard error of mean. The demographic characteristics and metabolic profile of the two groups were compared by students' t-test. Adiponectin mRNA levels within the groups were compared by paired t-test. Results. The serum adiponectin level was significantly lower in obese as compared to non-obese. the visceral and the subcutaneous adipose tissue adiponectin mRNA levels were also significantly lower, respectively, in obese as compared to the non-obese. The VAT and the SAT adiponectin mRNA levels did not differ within the groups. They were statistically almost the same. This table shows the demographic characteristics and metabolic profile measured in the two groups, that is the obese and non-obese. Here we can see the body mass index and the waist circumference demonstrates a significant and positive correlation. On the other hand, this table shows lipid profile, VAT, and SAT adiponectin mRNA expression levels of the two groups. Here we can see the triglycerides, the VAT mRNA, the SAT mRNA, and serum adiponectin levels is significantly correlated. The VAT and the SAT adiponectin mRNA expression levels in the two groups here shows in non-obese, that is the first one, A, and obese, that is the B. They are all postmenopausal women. The values are expressed as mean and standard error of mean and compared by paired t-test. Both the body mass index and waist circumference demonstrates significant and positive correlation. Further, the VAT adiponectin mRNA level shows significant association with both triglycerides and high-density lipoprotein after adjustment for age and BMI, indicating significant association independent of obesity. This table shows that age does not correlate well with any of the variables. However, the body mass index and waist circumference, they show positive correlation with triglycerides and negative correlation of serum adiponectin, VAT and SAT adiponectin mRNA levels was also observed. Several studies have demonstrated that adiponectin enhances insulin sensitivity, increases fatty acid oxidation and glucose uptake, and suppresses hepatic glucose production. The studies show that adiponectin enhances insulin-stimulated IRS1 tyrosine phosphorylation and AKT phosphorylation, providing a mechanism by which adiponectin increases insulin sensitivity in the cells. In the present study, we found significantly serum adiponectin concentration in significantly lower serum adiponectin concentration in the obese as compared to non-obese. In contrast, reduction of body weight in obese subjects increases serum adiponectin concentration. The present study, the VAT and the SAT adiponectin mRNA levels show significantly lower in obese as compared to non-obese. However, the study did not show significant difference between the visceral adipose tissue and subcutaneous adipose tissue adiponectin mRNA levels either in non-obese or in obese, though in both cases it was lower in SAT than VAT. Additionally, this study showed significant and independent association of VAT adiponectin mRNA level with triglycerides and high-density lipoprotein when adjusted for age and BMI, thus suggesting its potential modulatory role in lipid metabolism. Summarizing, we find an association between the visceral obesity and metabolic syndrome, but the pathophysiological mechanisms which explains this link is not completely understood. Adipose tissue is an important organ to produce various adipocytokines, which are involved in inflammatory pathways. Development of insulin resistance with increasing adiposity suggests that these adipocytokines may be important in insulin resistance. Adipose gene expression of various pro-inflammatory cytokine is elevated with obesity, whereas the expression levels of anti-inflammatory adipokines like adiponectin lowered in obese individuals. The present study found a significant effect, especially of adiponectin mRNA expression levels on triglycerides and high-density lipoproteins. Moreover, the study also found significantly lower visceral adipose tissue and subcutaneous adipose tissue adiponectin mRNA levels in postmenopausal obese as compared to postmenopausal non-obese. The study concluded that there may be a significant role of omental adipose tissue in obesity, which inversely and directly regulates metabolic risk factors like triglycerides and HDL, respectively. This is a research paper published from this work in the journal Hormones. The paper was Adiponectin mRNA in adipose tissue and its association with metabolic risk factors in postmenopausal obese women. To conclude, the study concludes that there may be a significant role of omental adipose tissue in obesity, which regulates the metabolic risk factors, triglycerides, and HDL, respectively. BMI and waist circumference show independent and significant association of bad adiponectin mRNA with triglycerides and HDL. This suggests its potential modulatory role in lipid metabolism in postmenopausal obese women. Thank you for your patient hearing.